and Max. So go ahead.
So yeah, it's my great pleasure to introduce Max Aleman today. So Max is originally from South Florida, but he decided it was too warm down there, and so he came up and got his bachelor's in biology at Franklin Marshall College here in Pennsylvania. After that, he went back, started for a little bit of time at UCF, where he was awarded an NSF GRFP, but decided he really loved the cold weather, and so he decided to transfer back up here to Penn State. So he's been here for the past 4 years. So as I said, he's been awarded a GRFP. He's also recently gotten an award through the Northeast SARE project or program. And so he's going to be talking today about some of his work looking at the microbiome associated with blotch of mushroom, and he'll tell us about how important mushrooms are for those who don't know to the economy here in the state, but also for Penn State in general. So Max, do you want to?
Thank you so much. Hola y buenos días. Hi and good morning. As Kevin said, my name is Max, and today I want to talk to you guys a little bit about my first chapter, my chapter 1. And so just to kind of give you guys a little guiding forward what we're going to be talking about, right? I want to introduce, as Kevin kind of mentioned, the mushroom production system, and specifically why mushroom production is so important here in Pennsylvania. Shout out to the Mushroom Research Center. It's a great resource. Yeah, hey, someone knows about it. It's a great, resource, and it has great questions. The mushroom system is a really interesting system. And I'll sell you on it, I promise. So then we'll go and we'll talk a little bit about the bacterial blotch disease, which actually should say disease complex, but I will talk about that in a moment. Then we'll talk a little bit about my objectives for the work, and then we'll talk a little bit about the methods insofar as the work that was done by the Bull Lab, which I kind of piggybacked off of and took their DNA from their mushroom communities and then did metagenomics on it. And I'll explain that a little bit more as we get there. And then we'll just talk a little bit about the results that I have today. So then let's get into the fun and exciting general mushroom production. And I'm not going to make you guys remember all of this. So let me just quickly break it down. There are 4 major parts of the mushroom production system. And the first part here is in 2 phases. And the first part is about composting. And this composting just kind of results in available nutrients for the fungi during production down the road. And the second phase of composting is actually kind of important because it's where pasteurization occurs. And this is typically where most pathogens in the system are removed. However, a particular pathogen that is present and persists is the Pseudomonas pathogens. So like Pseudomonas thalassei, which is a common mushroom pathogen and is the one that we'll be talking about a lot in this paper, in this presentation. So they persist throughout this pasteurization. And this is kind of the major time when bacteria are removed from the system, but you don't want to remove all the bacteria or all of the microbiomes and microbial communities because some organisms in this system, such as Pseudomonas putida, for example, assist in the formation of these mushrooms. So it's kind of an interesting and delicate balance where you want a microbial community, but you want to limit the amount of pathogens in that microbial community that persist in this system. So then you get to the spawning, and spawning is just the inoculation of your compost with a substrate carrier. And typically you think of this as corn colonized by mycelium. I don't know if anyone has seen that process, but you dump it into your substrate and you kind of mix it around, and then it gets pushed into like this block. And it's really interesting. That's how the mushroom grow, is in that block. And then once you have, once the mycelia have fully inoculated your substrate, you're going to go ahead and add a casing layer. And this casing layer is another source of where bacteria are added into the system. Typically, you want to use something inert and somewhat sterile, but the sterility of this casing we don't have, to my knowledge, a fully sterile casing material that is efficient for the pinning of mushrooms. And pinning of mushrooms is just the formation. It's a little baby mushroom. I think they're very cute, and I didn't put a picture. I'm sorry. Actually, I think right here. Yeah, little babies. Okay, so once you have the casing, your mushroom will start to begin to grow through. And this is where you have the production and harvest. And production and harvest is also coincidentally where I'll be spending the most of this time on this in this presentation, because this is where bacterial blotch typically occurs. And the reason why bacterial blotch typically occurs is because mushroom need water to grow. And typically the casing traps a little bit of water within the system to allow for the mushrooms to grow. And during this time, if environmental conditions either leave the mushroom cap too wet for too long of a period of time, the mushrooms are actually just touching each other. This is where bacterial blotch can begin. And the symptoms can start as quickly as 4 to 6 hours after you've left the water on the mushroom cap for too long. So I keep talking about bacterial blotch. So what is bacterial blotch? Well, bacterial blotch is this that you can kind of see right here. And credit to Clearly Bull, because this is a beautiful picture and I've gotten so much mileage out of it. I love it. So the symptoms can be, or you can typically characterize the symptoms of bacterial blotch as brown lesions. They're typically 2 to 3 millimeters deep and it can appear water soaked. I like to say it's a slicky, slimy texture, and it's, in my opinion, very gross to touch. And the first characterized pathogen for this disease was Pseudomonas thalassae. And so further work after the first species that was discovered, we've found a lot more species of Pseudomonas that can cause bacterial blotch. And now we also know that species outside of Pseudomonas can also cause bacterial blotch. And this is why earlier I referred to it as a disease complex, which is a group of species that can cause similar symptoms in the same crop. So this is why we would call it a disease complex, is that there are more than one species across genera that can cause a similar symptomology to this brown discoloration. So I've talked a little bit about how Pseudomonas plays both a positive and a negative role in this system. And we talk about how you can't really control the bacteria that are in the system, and that in fact you need bacteria in the system to have good harvests. So when we talk about how we can control bacterial blotch, it's not about necessarily destroying the entire microbiome, but rather managing the inputs of the microbiome. And so one of the ways, the first thing that you have to do in order to manage a microbiome is kind of understand what that microbiome looks like. So the purpose of my work has been to, if you kind of look in this area, something that's particularly important is that you can see these disease mushrooms here, but do you see that guy right there and that guy right there and kind of that guy right there? Those are what we consider a healthy phenotype. And as you can see, you have a healthy phenotype very close to these disease phenotypes. And how these are grown, mushrooms will grow in these kind of clusters, and you will find asymptomatic and symptomatic mushrooms. So my interest and the interest from the Bull Lab has been, can we see differences in the microbial communities between these phenotypes? So thinking about how we manage, can we see if there's any specific members of any species that are present on this cap that might be affecting that phenotype? So it's kind of the premise for the work that I have. And so this can be broken down into 3 objectives, which are the 3 objectives that I'll show you results for. So the first one is to compare the species diversity of Pseudomonads between asymptomatic versus symptomatic mushroom caps. And this is because, as I kind of let loose in the beginning, species level identification is relevant for disease management strategies. Pseudomonas putida is a positive Pseudomonad, while Pseudomonas thalassae is a pathogenic. So we need to get down to that species level resolution if we're ever going to have effective management strategies. So the second aspect of this presentation, or whatever, the work that I'm doing, is to compare the species diversity. So not just looking at Pseudomonas, because that's a little bit of a scope to view. There might be other members playing a role, right? So yeah, I want to look at Pseudomonas specifically, but I also want to leave for the opportunity that there might be other species playing a significant role in the phenotype that we see. And then lastly, I want to determine which, if any, species were differentially abundant between symptomatic and asymptomatic phenotypes. So those are the 3 objectives. So quickly to touch on the methods, I told you that the Bull Lab were the ones that kind of kicked this off. And so what they did was they went into one of these growing facilities, and then they went around the facility and picked 2 mushrooms as a pair. So they would find a healthy mushroom and then an disease mushroom, or an asymptomatic and a symptomatic mushroom. And then they macerated, excuse me. For this case, the relevant community that we're looking at was the community that was washed off the cap. So they suspended the caps in water, sonicated off the bacteria, they extracted the DNA and then they did sequencing on that DNA, but they did 16S sequencing. So my work has been going and doing this whole genome shotgun metagenomic sequencing so that we can do the comparative analysis across these phenotypes at a species level, which is again where that's relevant. So previously, as I kind of talked to you all about, we found that at a 16S level, so if you can just get to about the genus level using 16S, that there are differences across phenotypes or between the phenotypes. So asymptomatic and symptomatic do seem to have differences in the relative composition of their communities, or at least the relative also the relative abundance of those species. And right there. And then you also can see that Pseudomonas, right? And to just orient you, because I realized I didn't, the cyan, the level of cyan indicates more that these taxa are found predominantly in healthy mushroom caps. Red indicates predominantly found in diseased mushroom caps. And then anything that's kind of like a gray or an ambiguous color typically means that they're found in both phenotypes. In this case, with the 16S data, we got to a soft preference of Pseudomonads to be more, you have to find different species of Pseudomonads in the asymptomatic mushroom caps. I was like, okay, cool. What does that mean at a species level? Because you can see that there is differences, but it could be that it's just abundance, or it could be that it's an actual species difference, species level difference. Okay, so I was like, okay, so I took that DNA and then I did whole genome shotgun metagenomics on that DNA, and then I sequenced it. And then I have now data, exciting data. And there's a whole lot in between that, but data, pretty data. And so what you're looking at here is the PCOA. The left side here is the PCOA of all species. So the returned communities that, or excuse me, the returned sequences, I filtered those based off of being 2% abundance or more. And then we generated a PCOA using that data. And what we find is that there are three kind of groupings that you can kind of see in the data. And this is actually supported by p-values in which species richness is different across the disease versus or the phenotypes. So you see different community richnesses in the symptomatic versus the asymptomatic. And that's interesting. So what we can see at least is that there are differences between the symptomatic and the asymptomatic. Okay, cool. At a whole level, looking at all of the genera in the samples, there is a difference. Cool. What does that also look like? So if you didn't like the visualization here, what's interesting about this visualization is you can also get a sense for the similarity of those communities to each other. And what we find is that there are these three kind of groupings, which is similar to what I just told you about before, which is, there's supposed to be another one there. Anyway, so we'll just go back one, that there are three groupings out here. So corroborates, the PCOA is corroborated by this relationship heat map here that will show, and what you're seeing here on that diagonal line is just the two samples being compared to one another. And so there's zero difference. And so as we get lighter, it's the difference between the species present in the community. So it's how similar are all of the species lining up to each other when you compare them across communities. So this is at a species level across communities. And so what you find is that here in this clade of where there's predominantly healthy or asymptomatic mushrooms, we see that these communities are pretty dissimilar. Right? You can see that they tend to find a lot of differences amongst themselves. But as we get closer to the disease, there is a reduction in the difference of species in the diseased. This is interesting, and I'll tell you why in a second, but I guess I can just tell you now. It's probably because Pseudomonas. So if you were to take all of the significant species, so then I did a DesiQ to pull out all of the significant species. So before I was telling you, we're just looking at all the species. What are the differences? Now I wanted to look at what are the species that significantly differ in abundance across the samples, or rather across the phenotypes, the asymptomatic versus symptomatic. And what we find, what you're seeing here is the number of species per genus that were found to be significant. So there are about 197 species that were found to be significant that were Pseudomonas. Now, the discerning eye might say there's not 197 described species or type strains. And that's true. A lot of these are species, like species with just an identified, so a single strain that was identified, and not necessarily a type strain. But what we can say is that at least for what we can resolve down to at a species level, Pseudomonads are significantly, are playing a significant role in at least how the communities are different between our phenotypes, right? Which is interesting. And then you can see that right there, that there is a difference in the richnesses observed in the phenotypes between the phenotypes. Okay, so then I'm like, okay, what are the Pseudomonads that are relevant? Or what are the significant Pseudomonads? So there's a lot of them, but I can't look at every single one. So what are the most significant is kind of what we said. And what we find is that the most significant, so this is fold change by either 2 or greater, and this is fold change by 3 or greater. You're going to ask me a question that's really relevant here, and I'm going to start wrapping up soon, but a really relevant question, which is, do these relate to the disease or the healthy? And I cannot tell you that right now. And that is my next step, is to correlate the changes in fold to the phenotype. Right now, this is a DESI-Q, so it pulls away the most significant species and then also gives you an idea of how enriched or unenriched they were across the phenotypes. I am not a data scientist, and the data management in a DESI-Q file to a PhyloQ file is a little bit harder than I intended. So there is a bit of a learning curve here. So what I can tell you is that when we look at Pseudomonas, that they are being, on average, the most significant ones. So the ones that have the lowest p-value are also the ones that are expressed in the highest fold change, which is kind of like makes sense, but it's interesting. So, okay, great. So the takeaway here is that there are, at a species level, differences between asymptomatic and symptomatic mushroom caps. That's where we can end. Everything else after that begins to get to speculation right now. But what I want to do after this is to introduce functional genomics. And the reason for that is because I think it'd be a great foil to have, okay, these species are changing, but now what are the functions? What are we selecting for between the symptomatic and asymptomatic? That might give us a more interesting idea of what these trends mean and what's actually interacting with each other. Because right now we have trends, and I can tell you like what's significant, what's not significant, and the differences, but what's driving those trends, I cannot right now attest to. So I want to end on, this is exciting. Thank you guys so much for having me. And I'd like to thank the Hockett Lab. I'd also like to thank the Bull Lab for the data and all of the help that they've given me. I also want to thank Samuel Martins and Laura Ramos Sepulveda. They are like the 2 people who were backpacking this data until they gave it to me. And then I also want to thank the NSF-GRFP and Microbiome Center. And I also want to thank back there, Rachel Hirschlag, sorry for pointing you out. She was the one that gave me the template for this presentation. I'm like really grateful. Thank you. This is a really nice, powerful presentation. Okay, that's all for me. Thank you so much.
Questions for Matt?